Okay, so this demonstrator was about the clinical data, sensitive clinical data that comprises the clinical phenotypical data for the patients and their imaging. And the team that was working on it comprised CRS-4, Maastricht University, Medical University Graz, University of Dundee, and BBMRI-ERIC headquarters. And the involved RIs were naturally BBMRI-ERIC and the Eurobioimaging. The CRC cohort is a relatively large colorectal cancer cohort that is spanning the whole Europe with more than 10,000 patients with different relevant diagnoses. And it contains phenotypical, clinical, and limited genotyping data. And for about half of the patients, it contains also histopathological imaging data. And this is where I saw this overlap with the EXA demonstrator. Plus, there is actually additional data contributed from Medical University Graz and MMCI, which in the course of the project ramped up dramatically from those 18 terabytes available particularly for the CRC patients contributed to the CRC cohort. But Medical University Graz has now available more than 1 petabyte of the colorectal cancer imaging. So that was also an interesting use case, which was not covered in the demonstrator as such, but we were considering it as an extension of the use case for the future. The demonstrator was split basically into 3 parts. One was the data cloudification, the other was integration with the EOSC-LIFE platform, and the third one was the privacy risk analysis for the whole slide imaging, because this is still a relatively open and not yet explored problem that is heavily impacting the guidelines on sharing the whole slide imaging for the research purposes. So for the image data cloudification, this was done in collaboration with the colleagues from Dundee, UBI. It was basically focusing on transforming the data from proprietary formats that are now heavily used in the histopathological world as the DICOM format for digital pathology or for histopathological imaging was never taken up that much by the community, initially because of the patent issues and later on simply because it was too late. And we tried to convert the data to an OME-TIFF format, which is a commonplace in the imaging communities, a standard within the EUBI, and integrated the OME-TIFF support in the AI pipelines. The clinical data cloudification was done primarily between CRS-4 BBMRI, Medical University, and Maastricht University, which was focusing on converting a custom model of the CRC cohort dataset into something that is more interoperable from the FAIR perspective. So converting the data first into an OpenEHR formalism from which it can be turned into different or mapped into different other ontologies and standards such as OMOP or HL7 FIRE, and then implement the actual conversion. The practical implementation of this conversion comprised mapping 55 out of 56 data nodes successfully, and finding appropriate positions in the hierarchies within the OpenEHR. And eventually, once this was mapped, we also did the mapping to the OMOP and HL7 FIRE, where some of the elements are relatively custom, or some of the data elements are so custom that they don't have direct counterparts in a well-established HL7 FIRE profiles nor in OMOP. So only those parts that were applicable were converted. And we have built a tool for the OpenEHR-based backend conversion for this. The next part was implementation of the dataset query service. This is based on the BBMRI federated platform, which is a next-gen discovery tool within the BBMRI ecosystem that is able to do a federated privacy-preserving querying of sensitive health data. And at the moment, it comprises of two systems, a locator and a finder, each of them working on slightly different data standards. So the locator works with the FHIR and finder works with OMOP, which is actually a good test case for us for the integration and for the conversions. And we have deployed connectors for both of the platforms, as we have a policy that each of the datasets should be connected to only one of those. At the moment, we have it for some reasons connected to the Finder platform, and I will give you a quick live demo of that. But as a long-term solution, we assume to reconnect it back to the locator service. And this is where the data is made queryable and the data is made accessible, because then once discovered through the BBMRI directory or through the locator or finder, you can request access to the pseudonymized data. Which requires all the steps necessary for transferring personal data, meaning signing the MTAs or DTAs, depending if you're asking for data only. And we're basically then, once these things are done, we're then supporting the export of the data using encrypted channel to cloud-based data processing services. So there has been a tooling developed based on the GH4 crypt system, so that we're able to transfer the data into S3 storage or SFTP storage. And it has been also evaluated with different clients like rsync over HPN SSH, rclone, or Minio S3 clients. We're relying on an envelope encryption scheme where there is a data encryption key, and that is wrapped in a key encryption key. And then these are then shared with the users on allowing access. And as I mentioned, the practical implementation is based on Crypt4GH. Sorry for messing up the name before. And it's available on the GitHub, and it's also available through Workflow Hub as a snake-make workflow. Last part of the demonstrator was the analysis of the risks of sharing whole slide imaging. The whole slide imaging, the histopathological imaging, is basically large-scale imaging in the order of tens of gigapixels typically. So the scans of those histopathological slides are in the order of magnitude of 100,000 times 200,000 pixels, and these are now widely shared under different conditions, from personal, considering it a personal data, to considering it almost anonymous data with just minimal risk if you look at the Chameleon or other international competitions. And what we aimed for here is to do the privacy risk analysis so that the communities working on this type of imaging can set up their guidelines for, or best practices for, sharing the data. Basically, we use the different models from supervised learning to unsupervised learning, and looked into a tile-based models versus the whole slide image models that were searching, that were clustering the slides. And the tile-based models were not particularly successful, but the whole slide image models were actually very successful, showing that the linking risks that are related to the sharing of this imaging, and not only sharing the same images, which are basically acting as a fingerprint, so that something that if it appears in different data sets, you can consider it a perfect linkage across the data sets. But even spatially related slides are still relatively identifiable if you have like consecutive or next to consecutive slides. And that still creates pretty high risk for sharing such a data as anonymous material in different data sets. So we have prepared a paper that is proposing some initial relatively defensive guidelines for sharing the whole slide imaging, and this of course basing it on the risk analysis we have done. This has been submitted to the Nature Communications. It has been the reviews have been quite encouraging, so we're currently in the first revision with them. Let's see how that works out. It's also available on medRxiv for those who are interested. The URL is provided in the slides. As for the next steps, we're still aiming to finish the Nature Communications paper. We're working on a dataset paper on the CRC cohort that would also describe all this work that has been done in the demonstrator, and finally reconnect the CRC cohort back to the locator where it should have been. And the CRC cohort is hosted by BBMRI, so from the long-term sustainability of the results that we have developed in this demonstrator, they are covered by the research infrastructure. Now, I will quickly, Helen, do I still have a minute or two, or am I?
You still have a minute or two.
Perfect. So I will do a quick demo once I get the right window for sharing. So let me quickly share this one. This is session expired. Nice. So let's enter again. This is the Finder tool that is part of the federated platform. If I start searching for patients here in their statistic, this is basically doing the privacy preserving federated querying. So no personal data is shared. It's not centralized anywhere as a platform as such. Of course, the CRC cohort in particular has been already pre-centralized, but this works on the distributed data sources by default, and you can search for different consistently across centralized as well as federated data sources, and you can search on basically any OMOP artifacts in here. So let's see how many female patients we have that were treated with bevacizumab monoclonal antibodies for the colorectal cancer. So I can run the query, and in a bit we should see. I'll go to the results directly. We should see the results coming here. You can see it's about 155 such patients, and you can send this. I mean, there are others who have responded, not just the CRC cohort, but because of the anonymization that is done in this, they're hidden. The results are hidden under the threshold. So there is just BBMRI here, and practically available, and you can go into the negotiator of the BBMRI where you can request access to the cohort, and then all the steps that I introduced in the first part of the presentation become applicable, because once the access is approved, I mean, here you have the authentication, you are filing the access request, and once it's approved, we can transfer the data in a secure way to an S3 storage or SFTP service. So that's it from my side, and if there are any questions, more than happy to answer those.
Wonderful, thanks Petr, and an excellent demo. Very brave to do that on a live thing. I don't see any questions at the moment, so while we wait for people to come in, I've got a couple. So I really liked your live data query presentation, and you're basically doing that request then for data access. And I was wondering what the latency on that is. Is that a fully automated process that somebody at the other end pushes a button? Yep. So
well, so there are. For some of for the locator, there is theoretically also possible to configure the local interface, the connector, in a way that it requires manual approval of each query, but that is not practically used for obvious reasons. So typically those biobanks and data repositories that agree to participate in the federated platform are then responding automatically, and the latency depends on the size of the local data, of course. So we have resources that are having millions of samples that are then a little slow at the moment, and we're working on performance optimizations in those, to be honest. But generally, as you have seen, for the finder and locator, the requests are typically responded within 30 seconds or a minute.
So that's actually really good. Time-wise. So we're struggling in scenarios as well with data transfer, where data transfer outside particular silos requires quite a lot of security considerations, and not only security but governance. And so did I understand that you were kind of physically transferring data outside?
Yes,
and it's being aggregated somewhere?
Yeah, well, for the CRC cohort as such, this is one of the exceptions within BBMRI ecosystem where the data is centralized. So there is a data transfer agreement between the data source and BBMRI-ERIC, and then there is an expedited access procedure available, which means we're only consulting the data sources for veto of the respective release of the respective portion of the data. But if they don't reply within a month, then it's considered as okayed. The normal operation mode is, of course, or operating mode is, of course, the decentralized federated, which is why we have the negotiator, because then that keeps track of the access process and monitors when, I mean, it allows initially biobanks and the biomarker resources and the data repositories to declare what they have available for a particular purpose, because that you find something in the federated platform does not necessarily mean that it's available for a particular purpose, because it can be narrow consented for some, or not that broad consented for a particular purpose. So you might get a response like samples data available but not accessible for the given purpose and can be only collected prospectively, for instance, as an example. And then once the MTA DTA process is done, which is then also monitored in the negotiator, then the data can be transferred. And of course, it depends on the policy of the given data source. If they only allow the computation to be sent in or sent to some trusted third party for the processing, or if they allow the data to be exported. But many of them, once it's under a contract and there are appropriate data protection clauses in place, they're perfectly fine with sharing the data for the export for a particular purpose.
So your governance is primarily sorted out for this. Very clear.
Yeah, and maybe just one comment, which is also visible on graphs nicely on CRC cohort or on CRC data in general, is they're now having really like petabytes of imaging data. And of course, they do have their internal compute capacities for their own research teams, but they don't have capacities that would allow the incoming computations at arbitrary scale. So then it's again about finding an infrastructure that is commonly trusted.
Yeah. Okay, really helpful. Thank you very much. I do have a couple of other questions, but I think in the interest of time, we will move on, and I'll encourage people to type their questions in the chat, and I'll